You, Jindal sir, Banjal sir, and the whole ESI organizing committee for giving me this talk. My topic is circadian rhythm in endocrinology. I hope this is I'll make it simple and do justice to the topic. This is really a tough topic and mainly a physiological topic. So to know the diseases, we should know what are the normal things. Then only we can interpret what is abnormal. So this is what I'll be talking, what is normal and what happens in some of the main diseases which we see in endocrinology. My talk today would be basically in parts like introduction, molecular mechanism of circadian control, interaction between the central and the peripheral clock, implication on the treatment, and conclusion. So coming back, circadian, what is circadian rhythm? It is derived from a Latin word circa, which means around, and diem is day. So circadian rhythm is something which is around 24 hours. Likewise, we have infradian rhythm, which is more than 24 hours. The best example is menstrual cycle. And we have ultradian rhythm, which is less than 24 hours. Usually it is for few hours. So circadian clock has evolved to enable the organism to anticipate environmental changes so that that organism can adapt and have physiological changes appropriate for that time. Circadian clock machinery is well coordinated and it has a transcription, translation, feedback loop. And it involves the mRNA expression, protein stability, chromatin state, metabolite production, its utilization, and turnover. This is what a basic circadian rhythm is. The basic, we already know that the melatonin increases in the evening when we go to sleep. When there is darkness, the highest body temperature is around at 7 p.m., whereas the lowest body temperature is early in the morning, say 4:30 a.m. And we have faster reaction time and coordination in the afternoon, and we have highest alertness in the morning at 10 a.m. So probably this is the reason we have organized this lecture at 10 a.m. for the highest alertness. This is a really tough topic. So coming back to the clock, the suprachiasmatic nucleus, this is a 20,000 neuron cluster in the hypothalamus. It has three basic properties: endogenous, temperature compensated, and entrainment. By endogenous, what is means that it has under constant condition, that without any external stimulus also, it will have its own oscillation, and which would be approximately around 24 hours, the circadian rhythm. Temperature compensated, that within a variation of physiological range of temperature, it has constant oscillation. And third property is entrainment or synchronization, that is by external clues, it can slightly change its oscillations. And what are these external clues? This is what we call as zigzag, the time givers. This helps to synchronize the body circadian clock with the environment. So we have like two types of this zigzag, photic and non-photic. Photic, that is derived from photon. So light is photic. The non-photic is the social interaction, meals, and exercise. How this non-photic exactly controls is not very well known, but it is believed that it acts via the intergeniculate thalamic nuclei and via secretion of neuropeptide YY, it controls the circadian rhythm. Whereas this photic stimuli, the light, this has retino-hypothalamic tract and it directly controls the suprachiasmatic nucleus. Not exactly controls, it actually gives signals. So, and what important thing here is that even if there is no perception of image, no perception of light, then also it will act. What is that means? Like if the person is even blind, then also he will have this photic response. And there is no cones and rods required for this. The ganglionic cells of the retina, this convey the signal. So even if the person is not having the perception of light, he will have this zigzag of this light. And these are more influenced by the short wave, which are the blue light, that have more impact on this circadian rhythm. So the circadian rhythm has been divided into like two parts. There is a central clock and there is a peripheral clock. The external factors, the Zitzibug, the photic mainly gives signals to the central clock, whereas this non-photic, they'll give both to the central as well as the peripheral clock. The central and the peripheral clock are also in coordination. They are also synchronous. They give signal back and forth to each other via hormones and sympathetic projections. So this, and the peripheral clocks, almost everything has its own peripheral clock, like the muscle, heart, liver, pancreas, intestine. These are also having their own circadian rhythms. So in a basic circadian rhythm, there is an activator and there is a repressor. Activator will lead to transcription, translation, and this, whatever the protein is generated, this will again give a negative feedback to the activators. So this positive and negative feedback is seen in almost everywhere, like in even bacteria and plants. In the mammals, we have period and cryptochrome. These are the negative givers, and the positive, which is the clock and the BMAL. So in every cell, body clock output are generated in cell autonomous manner via this loop, transcription, translational, feedback loop. This loop operates approximately for 24 hours of cycle, activating a rhythmic cascade of transcription and post-transcriptional event involving thousands of targets of genes. Central to peripheral synchronization provide means of organs and tissue to function with maximal efficiency. Approximately 10% of the genetic transcript have this circadian periodicity. So this is the basic. Here, as we can see, this clock and BMAL, this is a positive thing. It will influence the E box, enhancer box, and will lead to transcription and translation of this CRY and PER, that is spirit and cryptochrome. When these are synthesized, they dimerize with each other and they give a negative feedback to the BMAL. Also, these clock and BMAL also produces this REV-ER-B alpha. This has a negative impact and decreases the synthesis of BMAL. So this is the main thing which is happening in the circadian loop, and this is the auxiliary loop. For better synchronization of the circadian loop, this alternate auxiliary pathway is also there. Both works in synchrony. Then there is another thing, ROR alpha. This is a positive thing. This will generate BML1. So I'll just repeat this slide. This is the basic of circadian loop. The BML and the clock, these are the positive thing. It connects to the E box, enhancer box, and will produce CRY and PER. This will give the negative feedback. Similarly, this REV-ERB, this also will give negative feedback, and this ROR alpha will give a positive feedback. So this all thing approximately happens within 24 hours, like this synthesis and their degradation, then again their synthesis. This all takes around 24 hours. So this circadian loop is of approximately 24 hours. And then there is circadian endocrine crosstalk. The main circadian rhythm, the central, is controlled by the suprachiasmatic nucleus of the hypothalamus. Then there is paraventricular nucleus and the subparaventricular zone. This also work in synchrony. They give signals, like here it is shown like they are giving signal via innervation, via synthesis of CRH, melatonin to the adrenal. This adrenal itself also has a peripheral clock. By that peripheral clock, I mean that there is a circadian gating mechanism. The response of the ACTH is also altered according to the circadian rhythm. The generation is obviously altered, the response is also altered. Then this synthesis the glucocorticoid, and this gives again signal to the other peripheral circadian rhythms. Then we have external things like meals, exercise, all these also, like with meal, the glucose increases, the insulin increases, the metabolite alters. These give feedbacks to both peripheral as well as central. In fasting, this FGF21 and ghrelin increases, while in fed state, this leptin increases. This again gives feedback to the central thing. Coming back to the main topic, the circadian rhythm, in endocrinology. So in endocrinology, there are two types. Like there can be intermittent secretion without any tonic release, which is seen in LH, FSH, GH, and ACTH. Whereas for some hormones like insulin, prolactin, and TSH, there is a constant release, and on that there is a superimposition of the pulsatile release. So there is a tonic release, on that various pulses in various frequency or depending on the hormone, it is secreted. By pulsatile, they've given definition like pulsatile nature of the hormonal release is like 100% change occur within less than an hour. If this happens, then we call it as a pulse. I'll be mainly talking in topics of circadian rhythm in this corticotropic axis, thyrotropic axis, and metabolism. Due to shortage of time, I won't be discussing about the somatotropic axis, lectotropic, gonadotropic, melatonin, and changes with aging, and in neonate, what is the circadian rhythm, or in the pregnancy. Coming to corticotropic axis, there is circadian rhythm of the glucocorticoids. This involves the HPA activity, like the suprachiasmatic nucleus will control the release of CRH, then the ACTH, and then the glucocorticoid. Also, there is autonomic innervation directly via the SCN to the adrenal, and there is a peripheral clock, as already mentioned, at the adrenocortical level. Under non-stress condition, this oscillates both in circadian as well as ultradian manner. Ultradian, that is like there are 12 to 18 pulses of this glucocorticoid release in 24 hours. Then there is endogenous circadian clocks, that is this SCN, even without any external stimuli, will continue its circadian clock, and this SCN has an stimulatory effect as well as inhibitory efferents are also there to the pre-autonomic and CRH producing neurons in the paraventricular nucleus. And this is about the secretion. After secretion, there is again a circadian clock at the receptor level. The glucocorticoid, these receptors are also rhythmically expressed in the various tissue, and they are subjected to acetylation and degradation. Also, this cryptochrome 1,2 protein has been shown to repress the trans-activation function of glucocorticoid. Like first level of circadian rhythm is at the central level, then at the adrenal level, then at the glucocorticoid receptor level, then at post-receptor trans-activational function is also circadianly controlled. Then there is proximal promoters of pirate genes, which are glucocorticoid response elements. So there is like a feedback also to this. This is a diagrammatic representation. Here we can see, like in the morning, there are pulses of ACTH, and so the cortisol is also high, and with the night, as in the evening, it goes on decreasing, and it's at the trough level at the midnight. Here what we can see that there is no tonic release. It is just the pulsatile release which is seen, and we can also see that there is inhibitory effect of the sleep on this ACTH and cortisol release, as well as other hormones like DHEA also show similar circadian rhythm as the ACTH and cortisol. And one more thing, what was observed, that if there is nocturnal awakeness, like arousal, even for few minutes, this cortisol peaks will increase. So overall, the 24 hours of the cortisol will increase in that person. This is shown for the disease, like we have Cushing's disease. By Cushing's disease means the ACTH dependent pituitary adenoma. So this upper one shows a single person, whereas this shows the mean with the standard error of mean for 10%. So this is normal rhythm, which has been discussed in the previous slide. In the abnormal rhythm, what we see is in the Cushing's disease, where it is ACTH dependent, the baseline cortisol increases, the pulses, the pulsatile nature is present, but the amplitude is decreased to a great extent. And in unipolar depression, which we also see like label as pseudo Cushing's, in this also the baseline increases, but the pulse are better maintained than the Cushing disease. Now this is about just the Cushing disease. What we see is in if that adenoma is at the pituitary level and there is ACTH direct secretion. If there is CRH secretion, then the things will again change. The CRH will stimulate the ACTH, and there may be hyperpulsatile this secretion, and the pulse may be better maintained in those patients. Then we have primary adrenal pushing, where it is ACTH independent. So you may argue that there won't be any pulses, or this rhythm may be more distorted, but it is not so, because there is innervation, and there is not just the circadian rhythm is not just controlled by this ACTH and all this, also there is innervation control. So the pulse is again slightly maintained, but obviously it is more disturbed than this one. Coming to thyrotropic axis, we can see here that as in the evening, there is slight rise of the TSH. It starts in the late evening. Actually, it is concordant with the melatonin release. And the highest TSH level is seen at early sleep. And in fact, when studies were done, it was seen that sleep deprivation increases this TSH mode. So this is the basic circadian rhythm which causes the increase in the TSH at early sleep, not sleep itself. In fact, sleep has an inhibitory effect on this TSH axis. If there is sleep deprivation, this increase of TSH is more in those persons. And now initially there was controversy about the T3 circadian rhythm, but now most of the studies have shown that the T3 rhythm is in concordance with the TSH rhythm. So decrease or absent nocturnal rise of TSH has been observed in a wide variety of non-thyroidal illness as well, including the hypercortisolism, hyperthyroidism, primary and secondary hypothyroidism, in poorly controlled diabetes. In this, that circadian rhythm, like in hypothyroidism, obviously the TSH will increase, the baseline will increase, but that rhythm will get disturbed, and that evening rise or the late night rise, what is seen is not seen in those persons. Obesity is associated with an increase in overall TSH secretion without alteration of diurnal variation. Circadian rhythm and metabolism. So circadian variation in glucose tolerance to oral glucose and meals, glucose infusion and enteral feeding, this has been seen, and is markedly higher glucose levels in the evening than in the morning. And there is diminished insulin sensitivity, decreased beta cell responsiveness in the evening, and this also correlates with the concomitant GH secretion, also decreased glucose utilization by the muscle and brain. They also have their own circadian rhythm. So this decreases in the evening, and thereby virtue of this, all these things, we have higher glucose level in the evening. Insulin secretion is also a complex oscillatory process. Like pancreas also have their own circadian rhythm. Also there is ultradian rhythm. Every 10 to 15 minutes, the insulin is released. This is an ultradian oscillation, and this can range from 90 to 120 minutes also. Stimulatory effects of the sleep on the insulin secretion are mediated by increase in the amplitude of the oscillation, and there is deleterious impact of sleep restriction on this glucose metabolism. How this is controlled, like there is suprachiasmatic nucleus, and it controls the peripheral clocks, and also directly and via the peripheral clocks, it influences the PPAR, gamma alpha expression, the rate limiting enzyme, mainly the NAMPT. This again controls the NADH synthesis. This metabolites also controls the metabolism. Also the nutrient sensors, which causes the insulin release, AMPK pathway, SIRT1. This is also impacted by this circadian rhythm. And the consequences of circadian rhythm disruption, there can be increase in the metabolic syndrome, cardiovascular disease. Also, it has been it has been seen considered as a probable human carcinogen. And the meta-analysis of short sleep duration and obesity, there is a odds ratio of 1.89. Like with short duration, with the sleep deprivation, the obesity increases both in children and adults. The odds ratio is around 1.89 and 1.55 in the adults. coming to the implication of this circadian rhythm in the treatment, sleep disorder, shift workers, and social jet lag results in disruption of the circadian rhythm, resulting in diabetes and metabolic syndrome. Chronopharmacotherapy is the application of this time drug delivery to maximize the therapeutic benefit of that drug. The term may also refer to therapeutic utility of drug that specifically target the activity of this circadian rhythm. Like here, I have just put a slide to show that various antihypertensive, when seen in trial, that when they were given in the evening, have a better benefit. They show more night dipping, and overall in the morning also the BP was lower when these drugs were given in the evening versus when they were given in the morning. And coming to a recent thing, chronotherapy in men, this Uradia, this multi-center randomized double control trial, what they have taken is the night shift workers and the asymptomatic patients, asymptomatic person of relatives of type 2 DM. And they have seen that these melatonin and the exposure to bright light and food intake changes, all these can have some effect. The initial results are not very promising. They have seen only changes in the HDL, but still it isn't going on research. To conclude the session, central and peripheral clocks act in synchronization to the various environmental zigzag. Various changes of the circadian rhythm should be kept in mind while interpreting the reports and diagnosing the disease. Modern lifestyle compromise the integrity of this feedback balance, and targeting this may provide novel solution to improve the general well-being. Chronotherapy could also prove to be an effective therapeutic option. Thank you for your patient hearing.